Hi, we're here with the Sugar Science, and we have the pleasure of speaking with a number of physicians and scientists from City of Hope today to talk about the life of Dr. Arthur Riggs. He, as everyone knows, is well known in the field of type 1 diabetes. He was a world-renowned diabetes expert who developed the technology that led to the first human synthetic insulin for the treatment of diabetes, and he passed away on March 23rd after battling cancer. He was really a biotechnology pioneer and a longtime leader at City of Hope in Los Angeles. And his major scientific discoveries were achieved over more than half century of research, paving the way for development of the monoclonal antibody therapies that are really the foundation of modern treatments for cancer, autoimmune disease, blindness, and a host of other diseases. He was a huge force in many places and really delivered a lot of therapy in each of the ones that he touched. Specifically in diabetes, though, his breakthrough work really enabled mass production of insulin for people living with the disease. And today, synthetic insulin is used by hundreds of millions of people. We will also be showcasing a link where you can learn more about him in our promotional materials. And he was 82 years old. Today, we have with us Dr. Debbie Thurman. She is the director of Arthur Riggs Diabetes and Metabolism Research Institute at City of Hope. And she's going to say, a few words and share, her impressions of him as a scientist, his contributions, and as a person. So welcome, Dr. Thurman. Thanks for joining.
Thank you, Monica. It's my great pleasure to have this opportunity. The loss of Art is a loss to science, but also the loss of my mentor and my friend. I think one of the things when I like to reflect on what made Art so special to me was when I first joined him, I sat next to him at a conference. Art had the most remarkable ability to sense, like a sixth sense, that project or that innovation that was the one that should be accelerated. So across my years of working with bright, brilliant minds, Nobel laureates, National Academy Scholars, I have to say that never before had I ever encountered anyone with Art's sixth sense of what was to be a fundamentally important breakthrough. I love sitting next to him at the conference because I would witness him paying attention to the speaker, and then he would reach into his left shirt pocket, I guess his right shirt pocket, and he would pull out a little notebook and he'd scribble something down. He didn't do this very often, but I knew that when he did it, it was something that was going to be key and important. He didn't necessarily even seek the speaker out. He didn't come up to the microphone to offer advice. If the speaker wanted to come find him, Art was always very open and approachable. I think the one thing about Art that was so wonderful during these conferences was his ability and the intensity of which he listened. Unlike so many people, he wasn't on his phone checking his email, checking his texts, reading the news, checking the stock market. He just listened very carefully. His gift was to see the most straightforward path, like a laser vision. He could distill what he listened to rapidly. He could distill the abstract thoughts, the complicated data sets and diagrams, or the ramblings of a nervous young assistant professor that knew that they were in the presence of greatness, Art. And then at the coffee break, he would simply and yet clearly drill down to me over a sip of coffee what he considered to be the essence, the essential truth that he had derived from that, and what it could mean to not just the field of diabetes, but across the span of fields. He saw things with such an ultimate simplicity. And actually, that vision, that ability to see things with that ultimate simplicity is why he identified all of these fundamental breakthroughs across scientific disciplines. Art himself, to the unknowing scientist or to a lay person, Art came across, he presented himself quietly, humbly, always kindly. And he had a purity of love for science. And I miss him.
We are very privileged and excited to speak with Dr. Fuad Kandil, MD, Ph.D., and Chair at the Department of Diabetes, Endocrinology, and Metabolism at City of Hope. He's here to talk to us about his recollections of time spent with Dr. Riggs, as well as his science and their collaborative efforts together. Welcome, Dr. Kandil.
Thank you. I'm actually, correction, I'm currently not the chair of the Department of Diabetes and Endocrinology. I'm the chair of the Department of Translational Research and Cellular Therapeutics.
Okay, thank you. Thanks for the correction.
All right. All right. I have known Dr. Riggs actually for 30 years or over 30 years since I joined City of Hope about 31 years ago. And I have started to work with him very closely about actually 25 years ago, when I actually took over the directorship of the Department of Clinical, of the Diabetes and Endocrinology in general, both actually clinical and basic science, translational science as well. Came up with the idea of actually engineering cell therapy for diabetes and immune modification. And that when actually Dr. Riggs became very excited, and actually he told me at the time he wanted to see eradication of type 1 diabetes in his lifetime. Mm-hmm. And he is willing to put all his resources and commitments into making this true. And it became really a partnership from that point onward. We developed the islet cell transplant program, and without actually his support, that program would not have been established at City of Hope. We really expanded the diabetes program from being one small clinical and translational and basic science research into a big department first, and then 2 departments, and then a diabetes center, and then a diabetes institute, currently with 8 departments. So that could not have been done without the unwavering commitment from Art Riggs. But during my experience of interaction with him, I recognized 3 things about Art. One, Art the human, again, Art the scientist, and Art the patient in his last couple of years. Art the human, he never really saw a bad thing in anybody. He saws all the good things from everyone. And he focuses on the positives of every person and try to promote those as much as possible. He was very kind to everyone, very caring to everyone, cared for the well-being of every person that actually crossed his path effectively. And he supported the individual human growth. I mean, everybody has a capacity for something, he promoted it. Everybody has aspiration for something, he actually did everything he can to actually make sure that that person realizes his potential. As scientist, he was a very great listener, but a very great connector of dots, so that whenever actually a lecture is given, he would see where that could lead to. and he would pick up on the positive aspect of it and how that can be actually accelerated or promoted to achieve something that maybe the presenter never thought of. So he was very remarkable in his ability to connect the science from different disciplines and from different standpoint of views.
That's very needed in type 1 diabetes because it's so heterogeneous.
It's so heterogeneous. And again, he very early on recognized that for us to be able to cure that, actually, we needed to focus on beta cell health and, and beta cell expansion and replacement, but also he was very supportive of expansion in the immunology aspect, which is really beta cell protection and alleviating the immune system pressure on the insulin producing cells from the immune system. So he expanded the Diabetes Institute in diabetes immunology, actually initially by hiring Dr. Dave Fuzeng from Stanford to actually work on mixed chimerism protocols, and then by really developing a department of diabetes immunology and hiring multiple actually investigators in this area as well. So again, never really lost the focus about how we can make human insulin producing cells, how we can expand them, how we can actually image them so that we can actually know what is happening to these cells over time. And again, he never really wavered from the commitment of trying to cure type 1 diabetes in his lifetime. And I think actually the Diabetes Institute is stuck to that actually aim, is we actually are focused on curing type 1 diabetes. If it didn't happen within Art's lifetime, I think Art has actually cultivated the environment and put the seeds in the soil that will actually lead to eradication of type 1 diabetes in the future.
That's amazing.
Lastly, I think I would say a few words about Art, the patient. Even in his course of sort of disease over the last couple of years, he was always thinking of science and linking everything to what could be done scientifically. And I can tell you, even at his last hospital admission, he actually told Will Davis, who is actually a development director at City of Hope, to actually raise funds for one of the projects that actually aimed to impact the healthcare of type 1 diabetes in a significant way. So he was committed to that to the last minute of his life.
That's an incredible story and an incredible tribute. And, I thank you for taking the time from your busy schedule for sharing that. And I'm sure that those who listen to this will really get the flavor of the way he lived his life as a scientist, as a person.
And again, Art really is the maker, the pioneer of making human insulin in the lab. So that impacted the life of diabetics, particularly type 1, all over the world, because actually previous to that, animal insulin was not very easy to use because it has some complications associated with its use, and people used to actually acquire larger doses and have fibrosis at the site of injections and stuff like that. And in fact, he did that against two things that I would mention very quickly. Number one, when he actually thought of the idea and wrote it as a grant application to the NIH, the reviewers actually said, well, that's a great academic exercise, but it will never work.
Oh.
But within a year of that, he proved them wrong. And he actually had produced the human insulin in the lab. The second thing that he did actually, which is very remarkable, the gene of human insulin was not known at that time, was not isolated at that time. So from the insulin chemical structure, Art was able to deduce the structure of the gene. And then he and his colleagues, Dr. Itakura, were able to make that gene in the lab, inserted the gene into E. coli bacteria, make the E. coli bacteria the factory for production of human insulin. There is no way you could produce human insulin cheaply than actually using the bacteria to be actually to become the vehicle for production of human insulin. And again, he made a gene that is the first human gene that was made in the lab. And that was really the start of the biotech era and industry, is really making human insulin gene in the lab and pioneering the production of human insulin for the benefit of type 1 diabetes worldwide.
We're going to be speaking with Dr. Anna Wu. She's Ph.D. and chair of the Department of Immunology and Theranostics at City of Hope. She is a cancer researcher by trade, but her interaction with Dr. Riggs is significant and important, and she's here to share with us, a little bit about him and how they work together in the field of cancer research. Welcome.
Thank you. But first of all, I do have to say that there is not one of us who has not been touched directly or indirectly by Art's contributions to diabetes. And I just want to tell a couple of vignettes of just how profound that change has been in a very personal sense. When I was in grade school and high school, there was a boy in my class that had type 1 diabetes. I remember when he was diagnosed because he was getting skinnier and skinnier and just not well. And then they figured out what the problem was. And of course, he was on, insulin before human insulin. It was always hanging over him. And I remember he came to our 5th reunion for high school, and he arrived in a limousine and cape because he was living life to its fullest. And for good reason, because he died a couple of years after that. And then flash forward to when I first went to UCLA, and I'll come back to that timing in a little bit. And I had a graduate student in my lab, and she had an insulin pump. Mm. But she lived you'd never know that she was a type 1 diabetic. She lived a perfectly normal life. She was getting her Ph.D. She was active. She was athletic. And over the years, she married, and she had 2 children. That would not have been possible without human insulin. So that's how much the world has changed, just a couple of snapshots.
Yeah, that's a very important perspective.
Yeah. So even though I'm a cancer researcher, I know that he's touched so many lives in so many ways, but I also think it's important for people to know how many lives he's touched in terms of cancer patients. Because I started my career at City of Hope in the 1980s. We won't say how long ago exactly, but he had moved beyond human insulin and on to recombinant antibodies. And that, that was sort of his next project. Oh, what else can I make that might be useful? And the question that he and his postdoc, Kabili, posed was, well, can we make antibodies in bacteria? A much more complicated protein. They have 4 polypeptide chains, not 1 or 2, and just very complicated proteins. And they did it. And that really launched the whole field eventually of the antibody therapeutics that we have now. Herceptin, Rituxan, all of the checkpoint inhibitor antibodies we use now that are now mainstays of cancer therapy. That all came back from his simple idea of, well, what else can we make? Let's make antibodies. And so I arrived after, the initial invention of recombinant antibodies. But basically, that's what my career has been built on, is engineering antibodies for clinical use. And so he would have these fantastic ideas, and some of the rest of us realized, oh, wait, this really could be useful. And that's what caused us to, basically my entire career is based on Art's work on engineered antibodies. And so that includes, starting my career at City of Hope, working with the recombinant antibody group, translating radio-labeled antibodies into the clinic with colleagues such as Jack Shively, Jeff Wong, so many others at City of Hope. And that allowed me actually to be recruited to UCLA, where I was for 16 years, where I worked on developing these engineered antibodies for imaging purposes. But then I was recruited back 4 years ago, really to co-lead the clinical program. And I think that's what's so important about what Art did. He had such simple ideas, but in the big picture, they had such potential and now realized potential in the clinical world. So, he just had that a way of just seeing what was possible. So he was tremendous when I first arrived at City of Hope. He'd actually already moved on to DNA methylation and chromosome structure by that time, which is one of the reasons I was able to step in and take on the engineered antibody program. But I just wanted to say a couple of things. First of all, tremendously helpful in getting me going, giving me clones of heavy and light chain genes for human antibodies, all of the ingredients that I needed to take on the program and to really work towards making products that would help patients.
Very collaborative and nurturing, it sounds like, from most people we've spoken to.
Absolutely. He was just so open and easygoing and generous with his time and his ideas as much as with his reagents. But he also had moved on to other things. And I think this is just, Art's brilliance. He was always thinking about the next thing. His imagination had no limits. He really, we tend to second guess ourselves, some of those who are scientists, because we're very practical people and very realistic and very data driven. He was the opposite. He was just a thinker, and that's why he came up with these ideas. And even, to hear him, he gave a talk just a couple of years ago about some of the new work that he was doing on chromosome structure and gene regulation. It's like his mind was always at the cutting edge of science. It was always amazing to just listen to him. And I also really appreciated his style, which was very laissez-faire. It really was. He kind of encouraged that in everybody. He would just provide resources, but he would just, let you turn your ideas loose and do things. So, I think that was the way he thought about and did science. And I think that's the environment that he created.
Yeah. And that can really foster creativity.
Right. I think that's really, in City of Hope's genes, as they say, is that
great, perfectly
environment.
said.
Yeah, that environment, it's in our DNA, that environment of just fostering creativity, not you're really letting people explore their ideas, not fettered by, I've got to get, at least having the support, not just I've got to do exactly what this grant or funding organization wants, but to also, in addition, have the freedom to explore ideas and just see where they go. So that always has been a tremendous inspiration to me, just his very laid back, laissez-faire attitude. But it just, he was able to turn the ideas loose. And I think that's really, to me, what epitomizes who he was as a scientist. But, that's why it's so amazing to have both of these in the same person. This, free thinker, incredible free thinker scientifically, but also somehow just hitting on, maybe because he was such a free thinker and he had no preconceived notions about what you could or couldn't do. Maybe that's why he was able to make these seminal contributions in two completely different fields, where he has impacted the lives of so many people that have no idea who he is. Also because he was so, low key, laid back, et cetera. He was not one to toot his own horn or brag about what he did. He just kind of did his thing. And then by the time all the, exciting products came out and discoveries and therapies, he was on to the next thing and the next thing and the next thing. So just very refreshing
Yeah,
to have
maybe sort of a
conversations.
tap on his humility, which I've heard, a few times now, that he had a certain sense of humility that accompanied his brilliance.
Absolutely. I mean, you visited him, he's still living in the same house, a very simple ranch house out in Glendora. He wears the same, if you looked at him, you never know he was, a gazillionaire or how much he's contributed because he
Right.
just show up in looking the same. Um, he was just a regular guy. And yet, if you just sat down in his office and he kind of look off in the corner and he'd say, what do you think if, and he would just come up with these ideas. So truly a gift to all of us.
Yeah.
Oh, he really was. It's truly a gift and an inspiration. I hope we can keep that sense alive, as we continue, to try to honor his legacy. I hope we can continue to honor and adhere to the, I wouldn't even say principles because he was very non-principled, to his
Maybe the tone,
his
the tone that he set.
yeah, absolutely, absolutely. So yeah, I miss him terribly. He really just was like grandfather to everything that we do, father and grandfather to everything that we do. It's just it's hard to imagine, a planet without him. He's given so much and done so much and was just always there and thinking about the next thing.
We're here with Dr. Stephen Forman, MD. He's the director of Hematologic Malignancies Research Institute at City of Hope in California. And he is our fourth interview today. We're talking to all these different scientists who are affiliated with City of Hope about what Dr. Arthur Riggs sort of meant to them scientifically as a person, to the City of Hope institution. And so welcome, Dr. Forman, and thanks for taking the time to chat with us.
You're very welcome, and thank you for having me. Admittedly, I was a little unsure when I was asked to do this because it said this was the Sugar Science podcast. And I'm thinking, why do they want a cancer doctor to join them? And then I reflected a lot on what Dr. Riggs had done, who he was at this institution, who he is at this institution, and who he has been to me for a very, very long time. I actually met him when I first came to City of Hope many years ago, and was aware of some of the work he was doing in diabetes. But I wasn't directly connected to that, obviously, because my work is about cancer.
Yeah.
But he helped me with a project that involved transplant patients who were getting treatment with stem cell transplant from donors to try to cure the leukemia. And there was a particular complication that they would have that was lethal. And me and a bunch of other people have been focusing on how to fix that problem. And Dr. Riggs cloned a gene for a protein from the virus that was causing all the trouble.
Mm.
And gave it to me and said, see what you can do with this. So we realized that the protein he gave us was immunologically significant. And we showed that if you could mount an immune response against that protein, then you would do better than if you did not. And a very long story started with that. I'm reflecting on one story. We were being reviewed by the NCI, as a matter of fact, about this project. And we were all in a room together, investigators and reviewers from around the country and staff from the NIH and NCI. And they asked me what my goal was for this particular research project. And before I could answer, Dr. Riggs said to them, our goal is to eliminate this virus from ever being a problem in any patient undergoing transplant. And, I was somewhat taken aback at the boldness of that statement, lofty as it is. And there was silence in the room. I mean, because nobody knew quite, well, what do you say? Well, that's so over the top, Dr. Riggs. But it was my first experience with him in thinking really big. You know, there are little increments in science that we all try to make and help and do our part. And then there are people who make observations that are just field changing, life changing. And I would say Dr. Riggs, for me, has always been that type of person in the most understated way you would just never believe.
Yeah.
I can tell you that what came from that work was, frankly, the development of immune strategies to treat that infection, drugs to develop to treat that infection. And then with another collaborator here, we developed a vaccine that is very effective. And it all started with the cloning of the gene for the protein from that virus. So if you trace the lineage of that work, it goes back to Dr. Riggs' laboratory, I believe in 1981.
Wow.
So that was about cancer. And I think for him, it was about human suffering and what we as scientists and physicians can do to relieve that suffering. You know, it was wonderful to be around Dr. Riggs and to work with him, but there are moments when you just felt a little overwhelmed and unable really to stand shoulder to shoulder with him, because his thinking was big and beyond, but without the type of ego that you see that contaminates a lot of discussions with famous, smart people. This was not him in the least. I found myself listening to every syllable he would say, in large part because he didn't talk a lot. So when he did talk, you know, I hung on to every word. I'm not sure if you've had this experience, but sometimes when you're talking to somebody and they're not talking very much, you start just blabbering and filling in the space.
It does happen. And yes, I've done that myself. It is interesting that you're, you know, in this lineup of, you know, discussions, I'm hearing again and again that he really was a listener, and he was listening and sort of parsing through things through his lens of understanding, and really kind of being able to distill things down to actionable items. I mean, it's pretty amazing.
Yeah, I think my appreciation for him just grew over the years because I wasn't working directly with his laboratory. But if there was a scientific question I just wanted his thoughts about, I would go see him or email him or call him. And the interesting thing about it would be, you know, when you have a conversation with somebody and it's done, you figure the conversation's over. But sometimes weeks later, I would get an email or a paper sent to me saying, you ought to take a look at this, which meant that, you know, he continued to think about it. And I was always, you know, grateful and honored that he would do that with me. I never, you know, I'm sure we've all had the experiences of feeling not very bright around somebody else who is so much brighter. But the other thing that I think was so important to me to see, because it was in contrast to, you know, other humans who are well known, is that for him it was only about the truth and scientific truth. He didn't suffer fools. He didn't suffer people who just performed. He looked for the substance that was there. And I think you exercised a cautious and suspicious eye on those he felt were pretenders. And the world is full of such people. And I think we were blessed that Dr. Riggs was in our community and really set a standard and a style, never flamboyant, never really drew attention to himself. It was, as I said, the pursuit of scientific truth through hard work and thinking clearly. I was always glad when he was in a seminar because I got a chance to hear what he would think. Obviously, I got to know him better in the last years of his life, but yet the the talking about science really never, ever was far away from the conversation. And I felt very blessed to work in a place that he had chosen to work for so long. In some ways, it sort of validated staying in this scientific community, because people like him were members of this scientific community.
Right.
And I was grateful for that chance. Me and a lot of other people whom he touched. I was really just, I would say, a minor character in his scientific life, but grateful for any time I got with him, and will miss him terribly.
Well, I'm not sure. I feel it sounds like, from just listening to others in this community, that, you know, he really, he made the time for those he thought had good ideas. And I'm sure you were one of them. It sounds like you had a great partnership there. And it does sound just like he set an amazing tone at the City of Hope. So what a legacy. It's amazing.
So I can tell you that I think this is okay for this podcast. I'm sure there are other places that have other people like Dr. Riggs, but I remember the night that he passed away. And it was late. It was in the night that I was still here. And I before, going home, I just went for a walk, around campus. And, these days the campus is not as populated as it used to be because of COVID and all that kind of stuff. So I went for a walk, and I felt like I was bumping into him every place I went, every building, a laboratory building, the hospital, the grounds. Even in the quiet of the moment, I could hear his voice. And I realized that his presence here is going to linger for a very long time. And I'm happy to help perpetuate that any way I can.
So we're here now with Dr. RAMA NATARAJAN, Ph.D. She's the chair in Department of Diabetes Complications and Metabolism at the City of Hope. And we're hoping to just chat with her regarding her experiences with Dr. Arthur Riggs in the scientific realm, as well as the personal realm, and how he impacted her career and her science. Welcome, Dr. NATARAJAN. Thanks for taking the time to talk with us.
Thank you, Monica. It's such a pleasure to talk about art. So I've known and admired Art Riggs ever since I came to City of Hope over 30 years back. So he has been basically my role model in so many ways. So his remarkable scientific discoveries, creativity, brilliance, innovative thinking are truly unmatched, I think. And interestingly, they were coupled with most gentle personality, filled with kindness, generosity, and compassion. Of course, all of us know that Art Riggs' scientific contributions are truly incredible. They cover not only fundamental discoveries, but also clinical applications. So the technologies that he has developed, all of us know, including insulin and humanized monoclonal antibodies, they continue to save countless lives of those afflicted with diabetes, as well as other diseases. And of course, in particular, we know that his work on insulin is absolutely incredible, significant for type 1 diabetes. Interestingly, his colleagues at Caltech, as I said quite often, that Art Riggs deserves not one, but 3 Nobel Prizes. So for his groundbreaking and pioneering work, and Art would smile in return and say, quote, he says, "My problem is that I get easily bored and my brain wants to continue exploring exciting new advances, RAMA. So, and actually he never stopped exploring. So, we know that his generous support of education and research in many places, including our own Artrix Diabetes and Metabolism Research Institute, has led to continued advances and worldwide recognition. But, I want to say something personally, because I think he has been the inspiration for my own research and my own prominence in this field, particularly our work on epigenetics related to diabetic complications and metabolic memory. So it was so heartwarming to hear him say several times, he, the renowned expert in epigenetics, he used to say that I basically started the field of epigenetics in diabetes, starting from some of our earlier work in 2004. And it was just so fantastic. Every time I showed him some new data or discussed a new project, his keen interest and enthusiasm, his eyes used to light up. And this would and this is just not with me, other researchers. This further enhanced my motivation. And in return, like a little kid, he was always so eager to show exciting new results from his own lab. And I really cherished those research exchanges over the years, which kind of, increased my own passion for research. And his passion for knowledge and discovery, truly infectious. And this kind of inspired all of us to continue thinking outside the box, which is something he loved to do. It was always impressive that how this man doesn't need to work a single day in his life, but he used to sometimes come to work way ahead of other people in the building. So I think that was like a motivation. In fact, he once jokingly said he's pretty out of touch with writing and applying for grants for funding. I laughed and responded. I said, Art, you don't need to apply for grants because you have been given grants for so many years. And so, it was always something we used to joke about. And apart from his endowments, I think we all know he was the anonymous benefactor supporting so many projects, including something called the Excellence Award on our campus. It used to give bridge funding for investigators in between two grants on those that scored a good percentile. And he also supported several initiatives for grants between City of Hope and UC Riverside and City of Hope and Caltech. So these were the two institutions that he did his early training in. And, a little bit of kind of the fun part. So he used to love discussing his vacations, which were always somehow clubbed with charity for wildlife preservation. So he had his quiet hideout in the mountains, which many people, I think this was one of the few luxuries. He never had any luxuries, but that was the only thing that he set aside time for every year. And his immense love for mountain biking. In fact, he used to say, he says, this crazy hobby of mine is probably kill me one of these days. But, and of course, the Laker basketball games, it was so refreshing to see him. One day he was jumping down the steps. I said, where are you hurrying off to? He says, a little secret, I have to run home to see the Laker playoff games. So he used to, I think his wife said he used to even tape those games and watch them later. Which was, of course, we used to always talk about the scores, Lakers scores. And he was also an ardent supporter of clean energy. He and his wife supported so many projects related to clean energy in California and other places. I think he was one of the first people I know who bought an electric car, even almost as soon as they were introduced. And he used to love showing off his Dodge electric car to me about his mileage and how much it was supporting clean air. So, there are so many little snippets that I would like to share with, but he has made an absolute indelible mark, not only in City of Hope, but everywhere. And the only thing I have been telling the future, the young generation, is that we should cherish and nourish his legacy, innovation, and discovery. And I think that's the way we can keep his name alive and his memory too. I think it was his humility that I wish many of scientists should appreciate, because I'm in so many meetings where there are all these top scientists who cannot stop boasting about the little that they have done. And here is this person who has done so much, and I never used to hear him boast about it. In fact, he used to say, well, I did this crazy stuff. Can you believe it? People after 30 years have actually proved that crazy theory I had so many years back. So, he put forward this loop extrusion theory many years back. It was experimentally validated. And this made him feel so good. So a true scientist in every sense of the word.
We're here with Dr. Jack Shively. He's a professor in Department of Immunology and Theranostics at City of Hope. And just offline, we've been discussing a little bit about his, relationship with Dr. Arthur Riggs, and they knew each other for 47 years. So he is well positioned to discuss Dr. Riggs and his contributions to type 1 diabetes, oncology, City of Hope, and probably most anything else. So welcome, Dr. Shively. Thanks for joining us. And we're happy to have, some of your impressions about what, Dr. Riggs contributed to the field of type 1 diabetes and oncology.
Okay, well, I'd be delighted to talk about some of those experiences with Art. You might say I was always a fly on the wall watching a lot of these things develop, and I even had a chance to get involved with a few things. And I'll just say at the beginning, in case I forget to mention it, is in 1975, when I first joined City of Hope as a postdoctoral fellow myself, I came with my wife, Louise, and one of the first things that we did when we arrived here was we had a chance for her to be interviewed by Art as a potential person to work in his lab. He hired her on the spot, and she continued working with him until the day that she retired, which was 2 years ago. And so in some of the books that have been written about that early work on insulin, they actually mention some of the things that she did. So I just wanted to give a quick plug to many people who loved and worked with Art for so long. If you think about it, that's a pretty amazing record to work with Art for 45 years.
Yes, that's a long track record. So, you're probably thinking like, wow, they must have had a good symbiosis and a chemistry working together in the laboratory.
Absolutely. And it just shows you that that's how loyal people were to Art. To know Art was to have a lifelong friend, and not just a friend, but a colleague and advisor. And also he had a mischievous side to him too. He liked to say outlandish things to get rises out of people. He was very athletic. And so he was the full person. You could do a lot with Art in different ways. It wasn't just science, but he mixed friendship, science, athletic activities, and then even some crazy ones like he used to have an outing with his whole department or people that were working with him once a year at Joshua Tree, and he would lead them on what we called the Death March. And it became famous that if you didn't have at least one death march with Art Riggs, you hadn't really, you weren't really a member of the research group at City of Hope. So it was an important part. And that just
what did that entail? I just marching in, hiking into the Joshua Tree?
Exactly. He would pick out a route and he would lead everybody without telling them how long and how hot and how much dehydration would occur during it. And I don't know if he did that on purpose, by accident, or whatever. But once it became enshrined as part of, you might say, the test of your mettle, you had to do it. The people who suffered the most, of course, were the new people who had never been in the desert before and didn't know that you needed like 3 or 4 bottles of water.
Oh, no.
And so I think at least on one case, somebody was helicoptered out. And that may be the time when Art realized that you can't just depend on people for figuring everything out. Sometimes they need to be guided.
That is hilarious.
In any case, that's just the personality of Art. He was really a forward thinker, very aggressive in his thinking of out of the box ideas. But when I first came to City of Hope, his main interest was in something that was really obscure at the time, and that was 5-methylcytosine. And he was telling, when I asked him what he did, he says, I'm working on exactly how the genome works. And he says, you probably think that you know that there's 4 bases in DNA, but really there's a 5th base, and that was 5-methylcytosine. And so I knew even at the beginning that he was interested in things at a more basic level, because, you know, who thinks about DNA at the base level and the fact that there might be an additional base in there that does something. And he was intrigued in terms of what that base did.
Yeah.
But that idea and the idea of, let's say, chromatin DNA regulation and how it could affect all physiological processes were always at the back of his mind. And of course, in those days, things like DNA sequencing and so on were just getting started. And people were just starting to think about simple things like manipulating DNA. And it was those ideas that finally led to the possibility of, let's say, expressing a what we now call a recombinant gene in E. coli. And you probably, I'm sure you've heard this story many, many times. But when Art was conceiving of this idea, the whole world was in a titter, as you probably already heard, because they felt that if you put something like a human gene into E. coli, a bacterium that could be anywhere in your gut, or let's say even the sewage system, my God, there would be human hormones everywhere. We'd all turn into freaks and so on. So now we laugh at that. But that was a serious concern because nobody had ever done it.
Right.
And so, as you probably know, Art found a way around the problem because they had placed a moratorium on it. And so he found that he could work with Keiichi Takura. And I don't know if you've interviewed at Keiichi yet, but you have to.
I hope I can.
Yeah. So Keiichi was one of the few people in the world at that time who could make DNA. And think about that. Chemically making DNA was, you know, I was trained as a chemist, a biochemist. And when I heard that Keiichi could make DNA, I said, wow. You know, so back then, nobody really could do that or conceive of doing it in any reasonable way, synthetic DNA. Of course, now people make whole genes. We don't really clone anymore. We actually order the DNA. Can you believe that?
Yeah.
So you go, you know, from the 1970s up to the 2000s, and you go, that's how much improvement just in that one area has occurred because it's so damn important. In any case, so Art figured a way around. He says, actually, let's make a synthetic gene. And of course, this occurred through a conversation with Boyer. And again, I'm sure you've heard all this story many times. but he and Boyer became fast friends. That was just a lecture that Herb Boyer gave at City of Hope, basically talking about one type of plasmid that we had in those days. One plasmid, that's it.
Wow.
And one restriction enzyme. That's it. That was the only two tools that
What
they
year
had.
was that? Do you recall?
Well, it's going to be, somewhere between 1976 and 1980.
Yeah.
And my memory is just not that good anymore. I mean, these are the kind of things that you don't write down at the time. Oh, this was the day, the month, the year that Art and Herb talked, because we thought they were crazy. Okay? You got to remember this. We all had our jobs to do, our research to do. And
Right.
here were these guys thinking about things that basically sounded impossible.
Yes.
But they said, okay, we got these two tools. We can make a plasmid that goes into E. coli, and we can cut it with one restriction enzyme. And if you can make the DNA, you can actually make that particular sequence of DNA that could be cut by that one restriction enzyme. So, well, that's cool, because if you rely on natural DNA, that's let's say that you get from cDNA, you don't have any choice at this point in time to make it so that it could be cut, say, by the right restriction enzyme. So anyway, so they brought Itakura into the picture. And then Art said, because they immediately wanted to do insulin. You probably know this story too. Well, Art said, that's crazy. You can't expect Itakura to make two long chains of DNA like this right off the bat. Why don't we start out with something simpler? And he picked somatostatin. Okay, now I'm going to diverge just for a moment here, because one of the things that Art did through his philanthropy was he endowed a graduate program in the ground floor of our Beckman building, which is on campus. And on the floor, first floor, he embedded into the tiles the sequence of somatostatin as read out from a DNA gel.
Wow.
And every day, our students, professors, and visitors walk across those and don't know what they're walking on.
It needs a plaque soon.
Well, I told Art this, and he said, no, I don't want to spoil the surprise. I want to see how many people have the same aha when they go in there. So there was actually one of the dinners, I'm going to say maybe already 8 or 9, maybe 10 years ago. I don't remember when. We were honoring some of the things that Art did. And Herb Heinecker, who was one of the key people. I don't know if you know who Herb is, but he was a key person back then, working with Herb Boyer and then Genentech. He came out to visit, and we were reminiscing over those days. And Art hadn't mentioned it to him, and so I did. I said, there's something you ought to see in this new Beckman building. And he said, what is it? And I said, I don't know, maybe Art should take you over there. And they stopped in the middle of their meal. They got up, and they ran across the way to the building, came back about 10 minutes later, and Herb had the best smile on his face I'd ever seen on a person. And I asked him, I said, did you figure it out? He looked at me, he says, in a heartbeat, Jack, in a heartbeat. So those were the things, So back then, when we sequenced,
it's very fun. It shows a side of him, the prankster, but also kind of like wanting to share that the joy of discovery with others.
Yeah. And then, just again, another aside, years later, when we would have student visitors, and I love the ones that came from Japan that hardly spoke any English. These would be young kids just deciding on whether or not to go into science or not. And they would ask me to talk to them in the foyer of that building. And they would be standing right in front of these tiles that show the sequence of somatostatin as run from a DNA gel. And I would ask the students if they knew what was in front of them. And of course, they got off a plane from Japan. They're trying to adjust to time and everything. What kind of crazy question is this? And so I would assign each of them a base, A, T, G, C, and have them stand on it. And I say, does this help you at all? And then they started to look at it. And a few of them actually could figure it out, that this was actually a gene sequence embedded into the floor of the building. And so that was somatostatin. Now, everyone wants to talk about insulin, obviously, but really, the revolution occurred with somatostatin.
Yeah.
It
Yeah.
really did.
It set the stage, right?
Now I'm a protein chemist, so I'm going to tell you what's wrong with all of that. So it turns out that when you have a gene for a peptide hormone, especially one as short as somatostatin, it's rapidly degraded.
Mm.
Okay, so all of our peptide hormones are made as long precursor proteins. You know,
Right.
some of the famous ones are from the pituitary.
Mm-hmm.
Okay? So Art knew that, and so Art says, you know, we can't just have the gene for somatostatin and expect to get anything, you know, out of E. coli that makes any sense. We need to make it as a fusion protein.
Mm.
So when people talk about what Art did, they often gloss over that none of this would have worked if he didn't make it as a fusion protein.
Right. Yeah.
And so again, he relied on what he knew about the literature. and over at UCLA, there'd been a group working on beta-galactosidase, which is a very large. Do you know about beta-galactosidase?
Yeah, I do. We use it as a marker sometimes, or we did. I did.
Absolutely. So all of this work on beta-galactosidase, you might say the guys over on the west side there, meant that the gene was available. People knew a lot about it. Its expression was very good in E. coli. And so Art said, let's take a plasmid for somatostatin and put it into the bigger plasmid for beta-galactosidase, make it as a fusion protein. Well, then there's still a problem, is because who the hell wants somatostatin attached to beta-galactosidase, right? So that solved problem one. You actually got the protein out as a fusion protein.
A Frankenstein, yeah. Protein.
That's right. You might say that the beta-galactosidase was like the Mack truck that had attached to it like one extra little piece sticking out the end. And it was that piece that we wanted. And so he also knew enough chemistry to know that if he put a methionine residue, which is basically the residue we all know ATG codes for and starts most proteins, if we put them methionine residue between the two, we could then treat the fusion protein with a chemical called cyanogen bromide,
right, which
had become famous over the years for giving cleavage at methionine residues. And it actually will release the fusion partner as an intact protein without any problems. And so Art did that, and he got somatostatin out, and the rest is history. So he actually proved that you could use a synthetic gene, you could get it expressed as a fusion protein, you could release it by chemistry, purify it by our standard techniques, and so on, and show that the whole thing was feasible. So that really meant that they could go on to tackle something more difficult like insulin.
Right.
Now I have to tell you one more great story. Hopefully nobody's told you this story. So when Art thought about recruiting Itakura, who's a chemist, okay? He's a hardcore chemist now. Hardcore chemists, they need special facilities. They have smelly chemicals. They cause explosions, yes, fires, all of those things we needed to give to Itakura at a hospital. and so there was really no good chemistry facilities present. And so Art went to the guy who was in charge at the time, the director, who was Rockmill Levine, who was really considered one of the world's experts on insulin. And so of course Art sold the whole story to him as, we'll start out with somatostatin, but don't worry, we'll make insulin. Well, Rockmill couldn't say no to that. And then Art dropped the bomb. He says, yeah, but we need a place to do all this chemistry because we got to make DNA. So what they did is they took over wing 1 of the hospital here at City of Hope. So remember, this is back in the early days. And so there were patients probably not more than 100 feet away from this robust chemical laboratory.
Oh God.
Indeed, they had at least one major explosion that I remember. The entire hood was blown off the wall.
Oh no.
Nobody was hurt. It was just a matter of luck. But it was in that smelly, noxious area. And the patients complained, the nurses complained, everybody complained, what is going on over there? They had no idea that it was history in the making.
Amazing.
Yeah. So anyways, so those are the early days. And I try to get across just how everyone thinks that science is so neat and organized.
Yeah.
And we know exactly what we're doing. We don't take chances. You know, we're conservative. None of that was true.
No.
Just the opposite.
And most of the best science is done, with a heavy dose of creativity. And a lot of times that's met with resistance. And, I think some of the most successful scientists have to have that underdog mentality. Hey, I'm going to try this anyways. I'll show you. And so it sounds to me that he had a lot of those qualities, which makes for an excellent scientist. And the proof's in the pudding, because he was able to, not only contribute so much to the type 1 diabetes field with the creation of, the protocols for creating synthetic insulin, but also in the oncology field, antibodies, and all that he's contributed in the scientific realm, starting at City Hope, moving globally, really. So,
what you're looking at here is a true basic scientist, a thinker. He didn't have diabetes. He didn't have a friend who had diabetes. That was never really his goal. Although what he wanted to do was show that there was a new type of technology
right.
that could benefit in that area. And you might say everything came together beautifully. Even in the area of cancer research, which Art made major contributions because of development of antibody technology, Art was never saying, I want to cure cancer per se. When people asked him what this technology could do, you know what he said?
No.
It can cure all known diseases.
Wow.
And when he said that, I almost fell out of my chair because it was hyperbole to the max. But it really shows you that's how basic scientists think.
Yeah.
They're not at the level of what disease should I go after so I can make a lot of money or make a big name for myself.
Right.
What they're trying to do is say, what kind of tools can I put together to make something work that otherwise doesn't work?
And they also seem to ask one of the most important questions is, what's the mechanism? Right. That's always a key question. What's the iteology? Let's just not throw drugs at this and see if it works. Let's find out what is actually happening.
Well, let me just put it in a different way though, because what happens to most scientists is that they can only do what their toolbox allows them to do.
Right.
If you develop a new tool, it's very unusual that people will cite you and get excited about you, invite you to meetings and so on, but they love to use your tools and to solve the problems. And those are the scientists and the companies and so on that get you might say, the big press. But if it wasn't for the basic scientists who you might say are toiling away with developing the new technologies, the new tools, we wouldn't be where we are. And I think that's the way it should be, because once you go to that bigger picture of like, I'm going to cure this disease and that's my goal in life, you really have moved on to a different aspect, let's say, of the technology or biology. It's just as important, but it couldn't have happened without that basic thinking that Art put into things. And that's what he loved to do.
Yeah, that's really well said. And I think much of the field of type 1 actually stands on the shoulders of this giant in the field, Arthur Riggs. Thank you so much, Dr. Shively, for talking to us about him today and really kind of shining several spotlights on his excellent work, his way of thinking, and all that he brought to bear. So we're welcoming Dr. David Horn, Ph.D. He's the Vice Provost and Deputy Director at the Beckman Research Institute at City of Hope here in California. And welcome, Dr. Horn. I'm really interested to hear about your interactions with Dr. Riggs, your memories and remembrances of him, as well as maybe just sort of, some of your scientific interactions with him and collaborations. What impact did he make on you?
Well, I met Art a long time ago when I was a postdoc at Caltech. This was probably in the late 80s, early 90s. And I don't think he remembered me from that one interaction, but and so he reached out to me a few years later, actually, many years later, I should say, when I was a professor at Oregon State in the chemistry department, because they were interested in bringing on a chemist to enhance some of the chemistry aspect of City of Hope. And so I was contacted by him, and he was asked if I'd be interested in moving to City of Hope. And I knew about City of Hope because I was at Caltech, and I kind of said, yeah, it's kind of intriguing. I think I was at a time in my professional career where I wanted to change. I wanted to do something that was more translational. I was a full professor in the chemistry department, but it was very difficult to do anything that was collaborative. Because in many times universities, especially if you're in the College of Science, you're doing chemistry, you're doing a lot of teaching. There wasn't a medical school there, and so it made it difficult, very difficult, to do any type of translational research. And I professionally felt a little bored developing another chemical method, or sort of another variation of another chemical method, which is typically how the field of chemistry progresses. And so I thought there would be a very interesting opportunity if we could apply some of that chemistry to some of the medical problems that are going on in the clinic. So I viewed City of Hope as a very unique environment, if you will, because it had the hospital, and it was surrounded by, in close proximity, the research facility. So I got a sense there was a lot of interactions going on between the physicians who are in the clinic. What's working, what's not working, what are the unmet needs, and how I could possibly contribute to a better understanding. And my goal, which is still hasn't been reached, was to make a difference in terms of possibly raising the standard of care in a certain unmet need. And I think that was one of Art's, it's always been, I think, one of Art's goal is, he would say, I've always wanted to make a difference. And so in that respect, this was somewhat of a perfect place for me because of the translational opportunities, which are a lot greater now than they even were back then, because we have a lot more infrastructure that's been built at the time. And Art was the type of person that, he was director of the Beckman Research Institute when I was hired. And so he was the type of person that would be thinking continuously out of the box. And
every single person so far that we've spoken with has touched on that he really thought outside the box, and have said that about him.
Yeah,
quote unquote. It's amazing that every single person has said it.
Yeah, he not only thought outside of the box, he worked outside of the box, and he would encourage investigators and set up the right infrastructure to allow people to work outside of the box. And we made agreements because when I was at the university, I had indefinite tenure, which it's at the time. It's thinking, this is probably the last best job on the planet, because once you have indefinite tenure, it's covers your salary, everything. You have a job for life.
Yeah.
And so, where City of Hope didn't have a tenure, but that, I think, didn't weigh on me as much as the excitement of just being part of City of Hope, working with Art in particular, and trying to build a vision that I think we both shared. And so when we were talking about what's needed, first thing was, like, Art, we need a GMP facility for small molecules. Because
Yeah.
if I want to make molecules, I could do that where I am and send it to you. But I want to do more than just that. And he said, yeah, I agree. Yeah. Well, we could do the GMP facility. And then the other part was, okay, well, we need to hire an X-ray crystallographer because that's essential for any type of meaningful translational work that we need to do. And so those were kind of the main points, and they weren't even at that time written down in my offer letter. It was pretty much a handshake. And so that was how much I trusted Art. And sure enough, they both came true. We raised money to build a GMP facility, which was constructed in 2012. And then we hired an outstanding X-ray crystallographer in John Williams, who I knew because I was on his thesis committee, Ph.D. thesis committee, when I was a professor at Columbia University. He was a graduate student, so I kept in touch, and I was able to recruit him here. And so both John and I worked very closely with Art over the years. And so so Art had that. He had that ability. He had that charisma. Even though he was somewhat of a quiet person, he had a lot of charisma, and you could immediately tell that by just having one-on-one conversations and kind of the breadth of interest that he had, and the knowledge and the depth of knowledge that he had. And you heard all the stories about synthetic insulin, monoclonal antibodies. And yes, it's true that there are a few scientists in history that have shaped the clinical management of disease as Riggs did. I mean, few have made, in the precedent set by their research, such a lasting impact on the advancement of biomedical sciences.
Yeah.
I think that is just an absolute truism.
Mm-hmm.
And so when my interactions with Art, it became closer as I took on more administrative responsibilities, because I would also always sort of seek advice from him. And so his interests spanned a very, very broad range of topics.
Right.
It wasn't just diabetes. It wasn't just monoclonal antibodies. He had interest in mixed chimerism. He had interest in small molecules.
It sounds like he was very interested in the mechanism,
Protein.
but also in the Lakers and in mountain biking. And
those were some of his hobbies for sure. Yeah.
He was a really a person, a scientific person, and a person in the world. Seems like he was firmly planted in both.
Oh yeah, in his own personal interests, he was, he believed in a green planet, that we were marching toward their precipice with global warming, and so he cared a lot about that. he loved traveling. He loved traveling to China. I used to go to on trips with him to China to give lectures several times. Deifu was another colleague that was very close to Art that would go as well. And so he had a lot of broad interests, and he was just extremely humble about things. You know, he would never be a type of person that tells somebody what they ought to do. And he would give, you know, his thoughts on things, and he actually had a very funny sense of humor that extremely witty and somewhat dry at times, but
a good wit is always a sign of intelligence.
Right.
Yeah, I think, you know, it just seems to me that in talking with all of these scientists from City of Hope that, you know, he was so many things, but the one thing, I guess the two things that really stand out is that he was an excellent scientist, really interested in understanding science for science's sake and how it can apply to do more good in the world, but also just a really great human being.
Well, yeah, and I think, you know, he had a rebellious side, and so it wasn't that obvious, but I think everyone who achieves what the man has achieved, which is pretty much unmatched, you know, has to have a rebellious side to overcome some of the obstacles.
Right. When someone says, no, you can't, when you can't do that, they inside say, you'll see.
Oh yeah. And for sure, he's competitive in that way, but he's competitive with himself. And so it's not, you know, the kind of tension type. It's because before the Genentech work actually started, and Herb Boyer said, "Oh, we should do diabetes." He had already submitted a grant to NIH to do insulin, and he would often talk about that. And
what were the scores on that? Did they?
Well, yeah, I think it pretty much got triaged. And so, you know, I'm paraphrasing, but basically what it said was, "Oh, this is just an academic exercise that's going to take forever." And so they didn't fund it. And he still, I think, you know, even to this day, gloats over it, right? So, but you know, he, like you said, he proved them wrong.
Yeah, it fueled him.
Yeah, no, absolutely, it got him going. So, and you know, it's just quite amazing because he had done all this other work before he even attempted to work on insulin. So, and he, you know, he would talk about how that actually helped him to just jump into the insulin project and do it the right way. That if he didn't make somatostatin first, these mammalian hormones being expressed in the bacteria, that you know, he just wouldn't have had the insight, and he may have lost the race, or they may have lost the race to be the first one to produce insulin from bacteria.
I think he exemplifies the quote, "If not me, then who?" Right?
Yeah,
because
I mean,
it seemed to jump in to these really big problems and hard questions and just tackled them.
Right. And I you know, I think he is a man, you know, from the Enlightenment period where you're going to admire the man for what he does, not because of that he's being forced or pressured to do something. Then you start losing respect if you have to operate that way. Whereas he was certainly the type of person that wanted the freedom of thought, wanted the freedom to believe in himself and create new things. And that's exactly what he did.